Hey there, welcome to Medic. We'll be learning about basics of gram-positive and gram-negative bacteria. Let's get started.
Why do we stain bacteria? Why are there different stains like gram stain, Zillnianson stain? Well, because bacteria, they are colorless and they are invisible to light microscopy. So we use these colorful stains to visualize them and also differentiate them like gram positive, gram negative. And it also helps the clinician to determine if the organism is round, say a cocci, or rod-shaped, a bacilli. Now coming to gram stain, it is a differential staining method devised by Christian Gram. And to say in simple words, first you pour on the crystal violet stain. Now this crystal violet stain, it is a blue dye, and following which you wait for 60 seconds. And then you wash it off with water, flood with iodine solution, wait for 60 seconds, wash off with water, and then decolorize it with an alcohol wash. Finally, counterstain with a dye safranin, wait for 30 seconds, and wash off with water. Now at the end of this, you'll see that some bacteria are blue and some are red. When the slide is studied microscopically, the cells which absorb crystal violet and hold on to it appear blue, while those in which the crystal violet is washed off by the alcohol, that is when decolorization is done, they will absorb safranin and they will appear red. They are your gram-negative bacteria. So crystal violet, blue in color, and they are positive. Whereas safranin, red in color and negative. So you can easily remember negative, red. Now, why is there a difference in the staining technique? Why does one take up crystal violet and the other cannot, and it takes up safranin? Well, the difference is primarily due to the peptidoglycan layer or the cell wall. Now, animal cells, they have just the plasma membrane. They do not have a cell wall. While bacteria, there is a special layer acting as a protective mechanism, and it is also significant when it comes to designing antibiotics. Peptidoglycan layer. Peptidoglycan, as the name signifies, it is made of sugar and amino acids which are covalently bound to each other and which forms a stable cross-linked structure. This is a rough pictorial representation. Now, the enzyme which catalyzes the formation of this linkage, it is called transpeptidase. It is called transpeptidase, and it is located in the inner cytoplasmic membrane. So now the clinical significance with this transpeptidase is that the antibiotic penicillin, it binds to this transpeptidase and it inhibits this enzyme, so thus affecting the cell wall formation. As I already mentioned that it's clinically significant. So for this reason, transpeptidase, it is also called the penicillin binding protein. Transpeptidase is also called the penicillin binding protein. Now let's look into the structure of this gram-positive cell envelope, the gram-positive cell wall. You can see here it has a peptidoglycan layer and a plasma membrane. This plasma membrane is the simple one, which was composed of phospholipids and embedded proteins. Now our focus is on this cell wall or the peptidoglycan layer. Now you can see here that the gram-positive cell wall, it is very thick and there is extensive cross-linking of the amino acid side chains. Now remember this because you find a contrasting feature in this cell wall when it comes to gram-negative. Now the gram-positive cell envelope has an outer cell wall which is composed of complex cross-linked peptidoglycan. You cannot emphasize this more than this. So the inner surface of the cell wall touches the cytoplasmic membrane. The inner surface touches the cytoplasmic membrane. The cytoplasmic membrane contains proteins that span the entire lipid bilayer. The bacterial cytoplasmic membrane, it has no cholesterol or sterols as we have studied in animal cells. So we have certain important polysaccharides present in the gram-positive cell wall, such as your teichoic acid and lipoteichoic acid. This teichoic acid, it is important for serologic identification of gram-positive species. It is an antigenic determinant. Teichoic acid is an antigenic determinant. That's about the structure of your gram-positive cell envelope. Now, this is interesting. Gram-negative cell envelope. Pay close attention to its structure. So you can see here that the first feature is that the peptidoglycan layer, it's not as extensive and strong as in your gram-positive. It is fairly a simple cross-linking pattern. And you can also see that it has 3 layers. Let's not include the periplasmic space. The 3 layers, you have this cytoplasmic membrane is your first layer, which is surrounded by a peptidoglycan layer, which is your second. And in addition, you have the gram-negative cell. It has a unique outer cell membrane. Now, this is what we're going to talk about. The inner cytoplasmic membrane, it contains a phospholipid bilayer with embedded proteins, same as we saw in gram. Following which, there is a periplasmic space in which you can find a gel of proteins and enzymes. And after the outside to the periplasmic space, there is the peptidoglycan layer. Now, this peptidoglycan layer, it is thin and it is not extensive as in gram-positive bacteria. You can see that it has no teichoic acid or lipoteichoic acid as in gram-positive bacteria, but it does have a small helical lipoprotein. This is called murin lipoprotein. Now, this lipoprotein is important because it originates from the peptidoglycan layer and it extends outward to bind the unique third outer membrane. It's a binder, murin lipoprotein. So this last membrane, which is similar to other cell membranes, that it is composed of a phospholipid layer, phospholipid bilayer with hydrophobic tails in the center. But you'll notice that I've marked something known as LPS. So that's what makes it unique. It is the lipopolysaccharide. Now this lipopolysaccharide, when we zoom in, we can see that it is composed of 3 covalently linked components. First one is your lipid A, outer to it is your core polysaccharide, followed by the O-specific side chain. Now, O-specific side chain. This O-specific side chain are nothing but they are outer carbohydrate chains of 1 to 50 oligosaccharide units that extend into the surrounding media. So these differ from one organism to another and are antigenic determinants. So O-specific side chain are the antigenic determinants in gram-negative bacteria, whereas in gram-positive bacteria, we saw that teichoic acid are the antigenic determinant. So this O-specific side chain is otherwise called as the O antigen. How can you remember this? So O-specific side chain, O, outer antigen. So the center part is water soluble. Coming to the center part, which is your core polysaccharide. So this part, it is water soluble. And interior to this core polysaccharide, you have the lipid A endotoxin, which is highly significant. So it is a disaccharide with multiple fatty acid tails extending into the membrane. When bacterial cells are lysed by the efficiently working immune system, the fragments of the membrane containing lipid A, they are released into the circulation. So remember, when your bacterial cell is lysed, this lipid A, they are released into circulation. Now this lipid A is the one which causes fever, diarrhea, and things like that, and it can also be fatal at times, which is the endotoxic shock. Septic shock, which we'll be covering in a later video. Now, embedded in the gram-negative outer membrane are porin proteins. You can see here, porin proteins. So these porin proteins allow for the passage of nutrients. They are also unique to gram-negative organisms, as we didn't see them in gram-positive. So to sum up, in gram-negative, we have the cytoplasmic membrane, which contains the phospholipid bilayer with embedded proteins. Outside of it is a periplasmic space, which has a gel of enzymes and proteins. Following that, you have the peptidoglycan layer. It is thin and it is not as strong and extensive as in, you can't say strong, it is not as extensive in gram-positive. and you have this murin lipoprotein, which is derived from this phospholipid bilayer, and it helps to bind to the outer membrane. Now, this outer membrane, it is phospholipid bilayer, but it's significant as it has a lipopolysaccharide. Lipopolysaccharide, it has three components. The first component is your O-specific side chain. It is an antigen determinant. Copolysaccharide, it is water soluble. Lipid A, endotoxin, highly significant as when the cells are lysed, it is released into circulation and it causes fever, diarrhea, and even septic shock. Last, we have porin proteins which allow for the passage of nutrients. Now, gram-positive and gram-negative. Why are we learning about this? What does this mean clinically? So the differences between gram-positive and gram-negative organisms result in varied interactions with the environment. The gram-positive thickly meshed peptidoglycan layer does not block diffusion of low molecular weight compounds. So it does not block diffusion of low molecular weight compounds. Low molecular weight compounds can pass through it. So substances that damage the cytoplasmic membrane, such as antibiotics, dyes, detergents, they can pass through. So however, in gram-negative, you have the outer lipopolysaccharide containing cell membrane. This outer lipopolysaccharide containing cell membrane, which blocks. So it blocks the passage of these substances to the peptidoglycan layer, and it has a sensitive inner cytoplasmic membrane. So therefore, antibiotics and chemicals that attempt to attack the peptidoglycan cell walls, such as penicillin, lysozyme, they are unable to pass through. So interestingly, the crystal violet stain used for gram staining, it is a large dye complex. So as we saw in the beginning, gram positive takes up the crystal violet, but your gram negative, it takes up safranin. So the crystal violet used for gram staining, it is a large dye complex that is trapped. So your crystal violet stain is trapped in the thick cross-linked gram-positive cell wall, resulting in the gram-positive blue stain. The outer lipid-containing layer of the gram-negative organisms is, however, in gram-negative organisms is partially dissolved by alcohol, so thus washing out the crystal violet, and it allows the safranin counterstain to take. Now let's do a bit of morphology. We know that bacteria, they are in four major shapes. They can either be cocci, which is spherical, excuse that, that's not proper, and bacilli, rods, spiral forms, like they can be comma-shaped or S-shaped or in spiral shapes. Pleomorphic, they do not have a distinct shape. Say they look like jello. So what are the names? You have gram-positive and gram-negative. How do I remember them easily? How to easily classify it? So let's start that there are 7 classic gram-positive bacteria that cause disease in humans. 3 cocci and 4 bacilli. So there are 7 classic gram-positive bacteria which causes disease in humans, and they are 3 cocci. Of these 3 cocci, the ones which occur in strips are your streptococcus and enterococcus, while the ones which occur in clusters is staphylococcus. The ones that form strips are your streptococcus and enterococcus, while the ones which form clusters are the staphylococcus. Now, gram-positive, 3 of gram-positive bacteria, they are cocci. Ones which look like strips under microscopic examination, streptococcus and enterococcus, while clusters, staphylococcus. Coming to bacilli, so 2 of the 4 gram-positive rods, bacilli rods, okay, excuse this, bacilli rods. Cocci is supposed to be sphere. So out of these, out of these 4 gram-positive rods, 2 produce spores. Spores are nothing but spheres that protect the dormant bacteria from harsh environment. So the ones that produce spores are Bacillus and Clostridium, and the ones that do not form spores are Corynebacterium and Listeria. So coming to gram-negative, there are only two groups of gram-negative cocci. So both are actually diplococci. Diplococci, nothing but they look like coffee beans, like two cocci together, diplococci. Examples, Neisseria and Moraxella. And there is just like one gram-negative bacteria which is spiral-shaped spirochetes. An important example is Treponema pallidum, which causes syphilis. The rest of the gram-negative bacteria, they are rods or they are pleomorphic. So coming to the exceptions, mycobacteria. Mycobacteria weakly stain so they are weakly gram-positive, but they stain better with a special stain called acid-fast stain. So this special group includes organisms which causes tuberculosis and leprosy. Also, in mycoplasma, there is no cell wall, so it is neither gram-positive nor gram-negative. Summing up the differences. So gram-positive cells, they have two layers, inner cytoplasmic membrane. So gram-positive, they have two layers, the inner cytoplasmic membrane and the outer thick peptidoglycan layer, where you have 60 to 100% of peptidoglycan. And while in the gram-negative, you have three layers, inner cytoplasmic membrane, thin peptidoglycan layer, outer membrane with lipopolysaccharide. The gram-positive cells, they have a low lipid content. So the lipid content is low in gram-positive. Whereas in gram-negative, there is a high lipid content. Gram-positive cells do not have endotoxins. Remember, endotoxins are the ones which are released when the gram-negative bacteria is lysed. When the gram-negative bacteria is lysed and it causes fever, diarrhea. So it doesn't have endotoxin. And also there is not much of periplasmic space in gram-positive cell, but you have significant periplasmic space in gram-negative. There are no pore-in channels in gram-positive, while you can find pore-in channels in gram-negative. The gram-positive cells, they are vulnerable to lysozyme and penicillin attack, whereas gram-negative is resistant to lysozyme and penicillin attack. Feel free to leave a comment below if you want to share information or suggest any topics. And don't forget to subscribe. Thank you.